So, I'm going to talk about our study about a vaccination in two native sheep breeds with our recombinant vaccine. And among the alternatives to alternative, we find two potential options in the use of vaccines and the use of resistant breeds against parasites. So here in this experiment, we have used a recombinant vaccine against Teladorsagia circumcincta, which had previously conferred protection in tertiary cross lambs, but it didn't confer protection in the younger lambs, and it had some individual variability. On the other hand, we have two native sheep breeds in here where we are in the Canary Islands that has shown differences in resistance against Haemophilus contagious, being the more resistant breed, the hare breed, and the more susceptible, the bull breed. So we have combined these two alternatives to compare the efficacy of the vaccine in both breeds at different ages. In order to do that, we carried out two trials. In the first trial, we had lambs of 6 months old and from both breeds, and we separated each breed in vaccinated and control groups. And in the second trial, we did the same, but with the 3 months old lambs. We followed the same experimental design as Nisbet and collaborators, where we vaccinated them 3 times, and then it followed with the trickle infections of the parasite, and lastly, we took stool, blood, and postmortem samples. So all of this led us to the following results. First of all, I would like to remark that we are going to follow the same color code for all of the graphics in this presentation, being the yellow and red line. Well, they correspond to the susceptible breed, being the red line, the vaccinated group of the susceptible breed. And the green and blue line correspond to the resistant breed, being the blue line, the vaccinated group. So here in this graphic of cumulative fecal egg count, we can see that there were some reductions of this variable in the 6 months old lambs when they were vaccinated in both breeds. But also we can see that there is an effect of the breed resistance when we look at the control groups, and if we compare it, there were significant reductions here. Then in this other graphic of the 3 months old lambs, we only saw a reduction, but no significant reduction, only in the resistant breed. About the nematode burden, here, if we look at the graphic of the 6 months old, we can see that there was a reduction when vaccinated in the resistant breed, but this reduction wasn't significant, and only it was significant when we combine the effect of the breed and the effect of the vaccine, if we compare it to the susceptible breed. In the younger lambs, this reduction that we saw here in the within the resistant breed is significant, and when vaccinated, obviously. So this could suggest that the vaccine is inducing this response here. Also, we look at the worm length, and there were interesting results. As you can see here, there is a reduction of the length, a significant reduction in the susceptible breed, and it reaches almost the same length as the resistant breed in the older animals. While in the younger ones, the reduction happened in the resistant breed, and it was significant. So this suggests that the vaccine is inducing, is reducing the worm length in the susceptible breed at older ages, and in younger lambs, it seems to reduce the worm length in the resistant breed. In these graphics follow the same pattern as the previous one, since they are related. So briefly, to summarize what I've said until now, the vaccine seemed to induce a protection in the susceptible breed and in the older animals, reducing length and eggs in utero. However, in the younger lambs, the vaccine seemed to be affecting the resistant breed, reducing several parasitological parameters. So all of these reductions are linked to the immune mechanisms that we also looked at, and we found that we observed the specific IgA against larval stage 4, where here you can see that there is a higher production of this IgA when vaccinated in the resistant breed of the older animals. And this wasn't the breed where the vaccine seemed to be reducing significantly the parasitology. However, when we looked at the correlations, they happened only in the vaccinated group of the susceptible breed, where the vaccine seemed to confer protection. In the younger animals, we didn't see any remarkable data, and neither we found any correlations, but there was a correlation in the vaccinated group of the resistant breed of IgA against L3 stage. So we think that this vaccine is inducing a response that is targeting the larval stages and is similar to the natural immune response of animals, where it's described that the IgA targets the larval stages too, regulating worm growth and prolificity. So we also observe globule leukocytes, which are mast cells, activated mast cells that we can find in the abomasal mucosa. We didn't see any differences in the number of globule leukocytes, but when we looked at the correlations, we found several negative associations in the vaccinated group of each age, where the vaccine seemed to be inducing the response. So also, we did find other negative correlations with parasitology, but we think that this is linked to the fact that the globule leukocytes are involved in the natural immune response of the animals, taking part of the rapid larval rejection. So our main conclusions in this study were that the vaccine efficacy depends on the age of each breed. The mechanisms induced by the vaccine are similar at different ages in both breeds. And this could imply that we could combine these two alternatives in order to obtain more data and keep improving the prototype vaccine, and also to integrate them in the future. Lastly, I would like to thank you for giving me this opportunity to give this talk, and also to the Modern Research Institute and my group at the Universidad de las Palmas de Gran Canaria. Thank you.
Thank you, Cynthia, for the very interesting presentation. We have several questions for you. Well, I start with the most popular. And based on which criteria you chose to test these age groups, 3 and 6 months?
Well, in the previous trial that Nisbet and collaborators did, they found that they had the vaccine seemed to work in this I think in the 6 months old lambs, but it didn't work in the younger ones. So that's why then we did the second trial, where we had the younger lambs.
And the vaccine was only injected once?
No. No, 3 times along the time.
Okay.
It was each 3 weeks. Every 3 weeks we vaccinated them, and then we infected
Okay.
later.